Yeah, thank you very much for the kind introduction. It's quite an honor for me to be here and give the first talk in this new seminar series. So I hope there will be many more talks and many more science going on this way. So I would like to talk about mathematical modeling of cancer. And in this talk, I will, of course, try to present my work, but the mathematical modeling of cancer is much wider than my own work, of course. So many, many important people have contributed many models and many results. I cannot cover all of them, of course, so I focus on what I know best, which is actually my own work. Now, the importance of mathematical modeling of cancer occurred to me even yesterday evening in the hotel. So back in November, I got one of my students approached me and said, well, Dr. Hillen, I know you work on cancer, and my wife has come down with cancer and was diagnosed such and such a way, and do you have any recommendations? And so I had to say to him, well, I'm not a medical doctor, I'm very sorry. So I can give you general advice, but I can certainly not make any recommendations. And so last night in the hotel, I got an email from the student, and he said his wife passed away. So it's really very sad, and I think we cannot give up. We need to continue to work on cancer. Even as mathematicians, we need to try our best and use our tools and mathematics to maybe help treatment. Okay, so in this talk, I will talk about three major things. Just remember, so the outline is a little bit in shaded colors. You cannot really see what's there, but I can tell you. So I will begin to talk a little bit about cancer, and in particular, what is the modern day understanding of cancer. So people are no longer assuming it is just a genetic disease. It goes much further and involves the microenvironment, the immune system, etc., etc. So I will talk about this a little bit. And then I have three main parts. The first one is on glioma growth. I really don't think you can see it. Maybe you can see it on the screen. So it's in glioma, which is brain tumors. And this will be the bulk part of my talk. It will maybe about 35 minutes or so. But then in the last 10 minutes, I would like to talk about radiation treatment, where we have done some work, and cancer stem cells. So each of them might be about 5 minutes or so of my time. Okay, so let's look at the cancer. So if you are really interested to work on cancer modeling or in this direction, I highly recommend the papers of Hanahan and Weinberg, The Hallmarks of Cancer. So there are two papers, and this 2011 is the next generation. It's the newest one. There was an earlier paper from 2000. And in those two papers, Hanahan and Weinberg characterize what is typical for cancer, what are typical hallmarks which make a tissue to be cancer and unhealthy. And so they summarized it in a number of, actually in two graphics. So this is the first 6 hallmarks from the original paper from 2000. So it's about sustained proliferative signaling. This is evading growth suppressors. This is, let's see, this one, enabling replicative immortality. So this green, brown, and blue, this has to do with signaling inside tissues. So what kind of signals are available, and the cells respond to the signal by growing or by dividing or by not dying or things like that. And then there are other characteristics. For example, the black one here is active invasion and metastasis. This, of course, is often the final stage of an invasive cancer. And if a cancer has metastasized, it's really, really hard to treat. And it's a bad prognosis for the patient. Now, the red one is inducing angiogenesis, also a very important process where a small tumor sends out growth factors, and then blood vessels come in and supply the tumor with blood, and also connect the tumor to the blood system. And then tumor cells can get into the blood and then metastasize and go somewhere else. Now, the gray one is resisting cell death. So through certain mutations, cancer cells, well, healthy cells, if they get into stress, they die. So if a healthy cell, for example, is radiated and it finds out it can no longer function, then it will die. But cancer cells might not die. They might still be able to survive in some form. So then the other 4 hallmarks, the new ones. So this one is deregulating cellular energetics. That's an important aspect, which is rather newly recognized. So typical cells use glycolysis to get energy for their metabolism. But in a cancer situation, they often a tumor does not have enough oxygen to do the glycolytic pathway, and they change the pathway, which is called the Warburg effect. And then they can still function on a lower energy level, but this gives them an advantage as compared to healthy normal cells, and this is also one way for tumors to invade. This one is avoiding immune destruction, and there's another one with the immune system, this one, tumor promoting inflammation. So these two relate to the immune response, right? So the tumor is recognized as a wound in the body, and the immune system responds to it and sends in macrophages, for example. It has now been found out that there are two types of macrophages, M5-1 and M5-2, and one are good and one are bad. So the M5-2s are actually good for the tumor. They provide the tumor with growth factors, they provide the tumor with angiogenic factors and with blood, for example. And so they actually, the immune system might even help to grow the tumor, not only to defend the tumor. And then this blue one is genome instability. So it is not only that tumor cells are mutated, they're also more prone to mutate even further. So they have an instability in the genome, and almost each replication creates a new type of tumor cells. So in a grown tumor, you have about 600 to 1,000 different mutations, which means you have specialists of all different kind of stuff. So you might have cells who do the Warburg effect like this, you have invaders, you might have cells who produce growth factors, you might have cells who compete with the immune system, etc. So what comes out of this, for me, comes only on the next slide, sorry. So here, first of all, the 6 first hallmarks, they have been all modeled with mathematical models in various contexts and for various cancers. What is now a hot topic is the genome instability and the interaction with the immune system. So these are current hot topics in mathematical modeling, as I see it. Yeah, as a note, as Bock said, all mathematical models are wrong, but some can be useful. So the only thing we can hope for is to get some useful contributions. Okay, so back to the hallmarks. So taking all this together, to me, cancer looks more like an ecosystem. So it has a microenvironment where it sits, it interferes with the immune system. Yeah, it has certain growth factors, it has many different cell types inside the tumor. So it's really more like an ecosystem. And so being in Mexico, I wanted to make advertisement for Canada. So this is Jasper National Park in the summer, not today, but it's quite pretty. But my point here is, so if you want to destroy this, maybe this is cancer, you want to destroy this, it is not enough to rip out all the trees, for example, because you have grass, you have birds, you have insects, you have many more things. So it is not enough to fight against one aspect of this ecosystem. No, this is not my idea. So Gartenby says, yeah, tumors are complex ecosystems. Hannah, Hein, and Weinberg, even in their paper, they mention that tumors are more than insular masses of proliferating cells. Instead, they are complex tissues composed of multiple distinct cell types that participate in heterotypic interactions with one another. And I think Merlot was one of the first one who made this impression public. So the tools of evolutionary biology and ecology provide new insights into cancer modeling. And so this is kind of my approach as well. Okay, so now let's look at the glioma growth problem. Now the problem with brain tumors is that you can visualize tumors quite nicely using MRI scans, but you don't get the full tumor. You get a gray mass in these scans, but you know this is not the full tumor. The full tumor has extended a bit more. And what is done nowadays is they take a 2-centimeter margin around what they see, and this is the treatment volume. So this is going to be surgically removed or treated by radiation or both, combination of those. However, the brain is a very heterogeneous environment. So it has white matter tracts which connect brain areas. It has gray matter. It has certain obstacles and membranes, etc. And it is known that tumors grow more in white matter direction and not so much in gray matter direction. So maybe this means maybe you need to treat a bit more than 2 centimeters in the direction of the white matter and maybe less than 2 centimeters in other directions. And this is exactly the problem I would like to discuss now. So it's about cancer spread in a heterogeneous environment of the brain. Now here's the first model in this direction by Swanson. It is a very famous model. It came out in 2000. And Christine Swanson has been very successful, and I think she's now at Northwestern University in the United States and has a large group where they do modeling on clinical data, data fitting, etc. So this model consists of a reaction diffusion model, so that's my first mathematical slide here. So we have a time derivative of the population density of cancer cells, and then you have a diffusion term and a growth term. So growth is just assumed to be exponential growth. And then the diffusion, you have a diffusion coefficient d of x, which depends on the type of material you are in. So here it's assumed you diffuse 5 times faster in white matter and a normal diffusion in gray matter. So this has been non-dimensionalized, etc. So the point is, it's 5 times larger in white matter than it would be in gray matter. However, the problem is that the white matter is a directed tissue. It is fibers. So the diffusion along the white matter tracks should be 5 times enhanced, but maybe in perpendicular direction it is even reduced. And so the assumptions of Swanson is the first important step, but I don't think they reflect the fiber structure of the white matter tracks. What we would rather have is something like this. So that the diffusion term is actually a tensor, which then in direction of the white matter is 5 times enhanced, and in perpendicular direction is just normal, or maybe even reduced. So what we need to look for, I think, is diffusion tensors, which give you diffusion or diffusibility in different direction with different values. And here's again another picture of the brain. So the white areas are the white matter fibers, and the dark black areas are the gray matter areas in the brain. And then here I overlaid a simulation of a tumor. So slowly I will get to where we do the simulation later in the talk. Okay. Now, there's a wonderful new modality in medical imaging, which is DTI imaging, diffusion tensor imaging. And this gives us exactly these diffusion tensors which I'm looking for. So this is a brain, and it is overlaid with a DTI image. And then the DTI image, in each pixel you have an ellipsoid. And this ellipsoid has 3 main directions, V1, V2, V3, and it has eigenvalues, lambda 1, lambda 2, lambda 3. And this tells you the isotropy in a certain direction. So if it's very cigar-shaped, it is on a white matter track and goes in a certain direction. If it's round like a ball, it is a gray matter and it has no dominant direction. So we do have data for patients for the for the direction inside a brain. So that's what we're going to use. Now, there's, as a side note, there are 2 representations of diffusion tensors. And I find this so cute, so I really needed to present it here. In particular, the peanut. So first of all, the ellipsoid, you have seen them in the previous picture. They arise if you take the inverse of the diffusion tensor and look at level sets of xt transpose times d inverse times x is constant. So these are also level sets of the n-dimensional normal distribution. So these are level sets of equal probability to find a random walker in this environment. Now the peanut arises if you met a unit vector into the quadratic form theta transpose d theta. And then, so let me show you the pictures. So here you can clearly see why this is called peanut. It just looks like a peanut. So I plotted 3 tensors in different anisotropies. So this one has 8, 1, and 0.2. So it's very highly aligned in the x direction. So this is more cigar-shaped than this one. And then also the peanut is more pinched off here than this one. So here so the peanut then tells you the mean square displacement in a given direction. So back to the diffusion tensor. So now this is a water diffusion tensor. So the measurement of the DTI is done on water. So you excite water dipoles to oscillate, and then you measure the resonance, and this gives you an idea about the diffusion tensor. So this is measured on water, and you can write it down in a representation using the eigenvectors like this. However, now we are interested in tumor cells. Glioma tumor cells are much bigger than water molecules. So they must behave a little bit different. The movement restrictions are different. And so Jebabdi and Swanson had a first idea how to do this, which is here. So in this representation of the diffusion tensor, they introduced a factor A1 of R, A2 of R, and A3 of R to kind of adapt the water diffusion tensor to a tumor diffusion tensor. But this is done rather heuristically, so they just tried something which worked. And R is a scaling parameter, so they played around with this R until they got a sufficiently good result. So let's look at their result. Here's another representation of the same kind of data, where now you can color code the directionality of the white matters. So green is tracks which go up and down, and let me see, red is tracks which go left and right, and blue are tracks which go into the screen and out. And then here's the enhancement. So that's the original picture, and this would be the enhancement used by Jebabdi with the factor R is equal to 10. So it's more pronounced than it was before. And then they used it on one patient where you have 6 pictures of the brain tumor, and then 6 simulations using the DTI data, and 6 simulations not using the DTI data. And I would like to point out only one thing. So here the tumor has a tip, and here in this simulation it has also a tip, but in this simulation it is round. So if you use the diffusion model alone as a prediction where the tumor might be, then you miss exactly the tip region here, and then that's of course the region where the tumor comes back very quickly. Now the question still remains how to really relate the water diffusion tensor to the tumor diffusion tensor. Jebabdi presented one way, but it was not physically motivated, and so maybe we can do better. And so here's a schematic which I developed with Kevin Painter from Harvard University. So now for the rest of my talk, I would like to do this, A, B, C, D, E, and F. So let me walk you through this a bit. Here's the brain image, and from this we get the DTI data, which is the ellipsoids. And then we want to infer something about the fiber structure of the tissue which is underlying, and then something about the movement of the tumor cells on this fiber tissue. And then we get a tumor diffusion tensor, and then we have a model which we can solve on the brain domain. So it's a long way to go, but I try to do this as clear as I can. So the first step would be to understand how do cells move in a fibrous structure. And nobody has allowed me so far to do experiments on human brains. So we use experiments on skin. So we assume that, well, skin consists of collagen fibers, which are indicated here. And we assume that the movement of glioma cells in brain tissue is similar than melanoma cells on the skin or in the skin. So we do skin experiments. So here schematically is a mesenchymal cell inside the skin. It moves along these collagen fibers. It can change direction. But what it also does, it changes the fiber network by cutting fibers. It releases a proteolytic enzyme and it's cutting fibers. And then it produces kind of a tunnel defect where the cell came from. So the next few slides, I want to develop a model for this skin cancer problem first. So here is an experiment done by Friedl and Wolf on fibrosarcoma cells. It shows one cell moving in such a fiber network. It is a rather short movie, but let me get it started. So you see, I think, a green cell, and then this gray background are the collagen fibers, and the green cell is moving around and is pulling and pushing on the fiber network. And it's also rather aligned to the fiber network. And then here in this slide, you can see where it degrades the fibers where it came from. So there's a little kind of a hole or a tunnel. Okay. And so in the next movie, I show you this in the context of a tumor. Now the screen, this movie is even shorter. So the screen will split in half. You get a tumor on this corner and on this corner, and then you see individual cells coming out and invading into the healthy tissue. So I probably have to run this twice. So this is short, isn't it? So stop it here. Yeah. And so you can make out individual cells which have moved into the healthy tissue and then other cells following the same trails more or less, which have been set up by the initial cells. Yeah, okay, so that's the process we are interested in. Okay, so now I would like to go to the mathematical model of this process. First of all, I need to model the fibers, the collagen fibers. And there are two ways to model fibers. You can have undirected fibers like collagen, or you have directed fibers. With direction, I mean so every way along the fiber, you can always decide what is plus or minus, or what is up or down, or what is active or passive. There are different notations. So, for example, microtubules or actin filaments would be directed fibers. However, collagen is undirected. So if you sit on a fiber, you cannot say what is up or down. It is symmetric. So each direction, theta, you have the opposite direction, minus theta, in the same way. Now the fibers are distributed according to a distribution function q of tx theta, so it integrates to 1. So this describes the probability of finding a direction theta among the fiber network. Now, if I have the fiber direction, I would like to compare the movement direction of the cells to the fibers. I would like to know if they go parallel to the fibers or perpendicular, or what they do. So my movement variable for the cells is v, or v, or the unit vector v hat, and capital V is a set of possible velocities of cells. So the important thing is it is a compact set and it has any direction. That's the only thing which really matters. Then for technical reasons, I need to lift the distribution Q, which is a distribution on the unit sphere, to a distribution on capital V. And I can do this by a scaling parameter omega in this form, Q over omega. So this is from the modeling not important, but it will show up in my formulas all the time. So you just should remember that omega can be computed explicitly, and it's just a known number here. Okay, so now the modeling of the cells I do through a transport equation. So P of Txv is the population density at time t, location x, and in direction v. And then this term alone is just the material derivative in direction v, or the directional derivative in direction v. It describes cells moving in direction or velocity v, and that's it. But now they do change direction, so I need something here on the right-hand side. And so this is the same left-hand side here, and now the right-hand side got those terms. So mu is turning rate, and so cells turn with a certain rate mu, and then they choose a new direction, and this is described by the integral term here. And the new direction is chosen from the fiber network. So if they change the direction, they look where are most of the fibers pointing, and then they go in the same direction as the fibers. So that's why we have the Q here as new directions. So Q over omega is a probability distribution for new chosen directions. Okay. Now, interesting, so this is an integral on V prime, but this depends on V hat. Hat is the unit vector. So you can actually take it out of the integration, and you can keep integral PV prime dV prime, which is just the total amount of cells in a certain location, which I call P bar. So you can write it a little bit more compact in this form, Q over omega P bar minus P. And then P bar is this. Okay. Now, I said these cells, they change the fiber network by cutting fibers, by degrading fibers. So this variable Q is time dependent. And I need another differential equation for Q. And due to time, I will not go into details how to derive the equation. I just show you that there is another equation for Q, which is basically an ODE. Yeah, so then we have a model which consists of two parts of an equation P for the cells and equation Q for the fiber network. And then Kevin Painter wrote a simulation for this, and I would like to show you now what happens here. So here's a square domain. The fibers are randomly oriented in random directions. Initial cells are randomly oriented, and then the cells start moving and start cutting, and other cells might find these roads and follow. And this leads to some interesting pattern formation. So first you don't see much structure, but then eventually you see these structures coming out. And then almost all cells will be moving about in these roads these networks, and not so much in between. And so now this a little bit resembles what I've shown with the tumor invasion earlier, that there are some leading cells and some other cells following the path. Okay, so now we have a transport equation and a Q equation, and this is all very complicated and too complicated to use for the brain tumor problem. So we would like to simplify it down. And for this, we use a multi-scale approach. So we are sitting here, kinetic model transport equation. What I would like to do is to derive a macroscopic diffusion-like equation. Also, what I've shown, the model of Swanson is here. It's a diffusion equation. But now my individual-based, cell-based model is up here. Now, there are several ways to do this. You can do moment closure, you can do hyperbolic scaling, you can do parabolic scaling. And in our paper, we worked all these out in detail. So here in this talk, I just show you the moment closure method and show you where this leads us to. So in the limit for the macroscopic cell density P bar, you get an advection diffusion equation, but it's a little bit strange as compared to maybe what you are used to. So first of all, you have a drift term where UC is the drift velocity, and it turns out this has a biological meaning. This is the mean fiber direction. So the mean fiber direction gives you the drift direction of the cells. It makes complete sense. Now, the diffusion term, you have two derivatives outside, and then D is a diffusion tensor, given like this, V minus UC and V minus UC transposed. So this is a dyad product, this is a matrix. You integrate it over the fiber distribution. So this also has a meaning, this is the variance covariance matrix of the fiber distribution. So the variance in the fiber structure gives you a diffusion term. And then you have the two derivatives outside. So it's a full second derivative of this tensor expression here. Yeah, so we achieved already something. Back to this picture, ABC. So we did we did A and B. These are DTI data. Now C is this function Q, which I defined. And from this, we can get a tumor diffusion tensor, which is the variance covariance matrix. So it's really only missing the last step to go from here to the full model. And one more ingredient I need is the von Mises distribution. So von Mises distribution is a normal distribution of the unit sphere. So here's the unit sphere, and then, a normal distribution has kind of a maximum somewhere. And you write it down like this. It has a Bessel function, I0 is exponential here. And then gamma is the given direction, here the maximum of this von Mises distribution. So I would like to use this to describe the fiber network. But since collagen fibers are non-directional, I need both directions. I need plus gamma, minus gamma. So I have a bimodal von Mises distribution like this. Now just assume I know the von Mises distribution, then I take this Q, put it in my transport model, and compute the variance covariance matrix, and I have my diffusion tensor. So here I do this for the von Mises. For the bimodal von Mises, you get a drift velocity of 0 because it's symmetric, and the diffusion tensor is this. So it looks rather scary, but it isn't. So these are Bessel functions, and each Maple and MATLAB and everybody has Bessel functions nowadays. So no big deal. And important is it has an isotropic part here, and I see I was missing the identity matrix. So here is the identity matrix, and then it has a directional part where gamma was the given direction. Okay. Good. So there's one more ingredient we need, which is the fractional anisotropy. So having a water diffusion tensor, DTI, and having these eigenvalues, eigenvectors, I can define an expression which is a complicated ratio of these eigenvalues, which is called the fractional anisotropy. So if it's close to 1, it's very anisotropic. If it's close to 0, it is very round shaped. And then you can plot on the brain domain, you can plot this expression at each point, and you get another idea about the directionality of the tissue. So red means high anisotropy and blue is low. So I think now we have everything to explain the whole procedure. So what we do now, we obtain the measured DTI data, and then we define Q as a von Mises distribution with a dominant direction E1, so E1 comes from the DTI data, and the concentration parameter K. So K is the level of concentration of the von Mises distribution, and we choose this to be given by the fractional anisotropy with a scaling parameter K, and then K is our adjustable parameter which we can use to fit data to the fit the model to patient data. So when we have Q, we can compute the tumor diffusion tensor as a variance-covariance matrix of this Q. And if we have this, then we can put it into the anisotropic diffusion equation of this form. Note the drift term has disappeared because drift is 0. So we solve this equation based on this information here. Okay, so let's see what we get. Again, this is a simulation done by Kevin Painter. So this is a healthy brain, but then artificially we introduced a tumor here and then we let it grow. And you see in particular the green areas, they really follow very nicely the white matter tracks. Stop it. No? Yeah. Right, so you see that the green is, what is this, 0.1 concentration. It is very fingery and follows very nicely the white matter tracks. And this is what clinicians tell us, this is more like a brain tumor looks like. It is not a round mass like a fist, it is more like a fingery spread. And the higher concentrations, so yellow is 0.02 and red is 0.004. These are very low concentrations. But then again, we hope to give clinicians this kind of information, and then they can decide, first of all, if they use it, and then second, if they use it, how they use it, and what kind of information is important to them. Okay, so another such simulation just recently done by my student. Now this is a real tumor patient. This is a data set from Cross Cancer Institute, and this person had a tumor sitting right here. And so my student wrote a simulation for this. And again, you see a very anisotropic growth of the tumor. The inner region, again, shows this kind of finger-like structure coming out. Yeah, so this is basically how far we got so far. So this is a purely two-dimensional simulation on one slice. So we have to get it three-dimensional, of course. And we also need to validate it to the data we have. So this is for one slice of one patient, right? But we have data for 100 patients, and they need to be three-dimensional. So there's still some more work to do. But I think in principle, we have a direction which we can follow. Yeah, so I didn't look at the time, but I think I'm right on, actually, where I want to be. Okay, so switching gears, let's talk a little bit about radiation treatment and then about cancer stem cells. And then I'll let you go for a late lunch or early lunch. So, yeah, okay. Radiation treatment. Now, I would like to explain the radiation treatment idea on prostate cancer because it is a very common treatment modality in the prostate case. And for prostate, you have basically two radiation treatment options. One is external beam radiotherapy, and one is internal. And internal is called brachytherapy. And I show you pictures, but they will not be scary, so don't worry too much. So here's a schematic of external beam radiation of the prostate. So here's a part of the human torso, and then you apply radiation beams from many different directions with an outline of the prostate. So here's kind of the outline is shown. And then if you show the overlap, then you get a high concentration in the red region where the real prostate is, and you get low concentration on surrounding regions. So with this method, you can really very sharply actually follow the contour of the prostate. It's quite amazing. The other method, the brachytherapy, you insert radioactive seeds into the prostate. So this is done a lot at the Cross Cancer Institute in Edmonton and seems to be very successful. So you just put radioactive seeds in there and you let them in there forever. But this seems to help quite a bit. Now, mathematically, what people are looking for is the tumor control probability. This is the probability that the treatment is successful. And there are several models for the tumor control probability. And I have been heavily involved in deriving several of them. So some models are based on a Poisson process, some are based on birth-death processes, others are based on branching processes. It turns out that these models give basically the same results if applied to realistic treatment schedules. So a realistic treatment would be having a patient coming in once per day for 6 weeks, but not on the weekends, and they get 2 gray per day as radiation. And this for 6 weeks, this is a typical treatment. And then you can think about if you give them 1.5 gray and maybe extend it to 8 weeks, or you give them 3 gray and reduce it to 4 weeks, what is better? So in this regime, it turns out that all the models more or less give the same results, and I show the results in a second, with one important difference. And the important difference arises in the number of cell types you study. So I call this 1 compartment and 2 compartment. What I mean here is 1 compartment means just you have 1 lump of cells, they're all the same, they're all the same sensitivity, etc. But now we have learned earlier from the Hanahan and Weinberg interpretation that a cell is very, very heterogeneous. You might have quiescent cells, you might have hypoxic cells. And so as an extension, we looked at 2 compartments where you have an active class, which is proliferating, and you have a quiescent class, which is not proliferating and not so sensitive. And now this does make a big difference in the tumor control probability predictions. So let me show you just the results without any details. So here I have 5 protocols, A, B, C, D, and little e and little f. And maybe let me explain one of the pictures first before I tell you what the treatment is. So let's look at C at the middle one. C is a 3 gray per day treatment. You get 3 gray per day. And then you see, look at these curves. These are 3 curves, the green, the red, and the blue. And they correspond to 3 different tumor control probability models. And as I said, these 3 different models using different approaches, they really do the same prediction. Also, what you see nicely, you have a step function which corresponds to the treatment days. So here's a day, here's a day, here's a weekend. This corresponds to 2 days. It's a bit wider. And then here's another day. Now, what you should see, a tumor control probability of 0 means treatment failure, and probability of 1 means treatment success. So with your treatment, you want to be somewhere here. Okay. Now, earlier you see a dashed line, also green, red, and blue. They're overlapping even more. And this corresponds to the 1 compartment situation, where you assume all tumor cells are the same. And this corresponds to the 2 compartment situation, where you have active and quiescent cells. And so there's a big difference, right, as you see. Now, the black line is where the treatment stops, right? So this is a standard treatment which is used in hospitals, and they stop right here. So it really depends what kind of model you use to make your prediction. If you assume all tumor cells are the same, then you are in the treatment success region. But if the tumor cells are not the same, then you are in the tumor failure region. And this could be one explanation why so many radiation treatments actually do not really cure a patient. So maybe they used the wrong model for predictions. Then the tendency for the other treatments, protocol A, protocol D, is basically the same thing, same story. So A is a 2-gray treatment, D is a 4-gray treatment, and these are hyperfractionation treatments where you give 2 radiation doses per day, so 1 in the morning, 1 in the evening. Okay. Good. So then finally about cancer stem cells. Now, cancer stem cells is another important class of cancer cells, and they describe cancer cells which have stem-like behavior. So stem-like means they are pluripotent, which means they can give lineages and they can differentiate cells to certain differentiation levels. They have they have unlimited replicative potential, so they can reproduce all the time. And they are much less sensitive to radiation or to chemotherapy. And then here's simulations on a cellular automata model done by Heiko Enderling. And just let's look at these 3 pictures here. So orange or yellow is a tumor stem cell, and then this is surrounded by the different shades of dark red and black. which are differentiated cells. And so a stem cell reproduces, it produces lots of differentiated cells, and they sit around the stem cell and they kind of cloak it off. And so then the stem cell doesn't get nutrients, and it doesn't have space to grow, and it just becomes quiescent. Now, if I start killing tumor cells here in this region, then all of a sudden I create space. And then the stem cell can start growing and produce maybe another stem cell. And this way you might get two seeds where you get then kind of the double amount of tumor. And then if I start killing more and more cells here, I create more and more space. And as a result, the tumor might become really big. So this alpha here is the death rate for cancer cells. So here alpha is 0, alpha is 0.01, alpha is 0.1. So I increase the death rate for the non-stem cancer cells, and I make a bigger tumor. So this is called the tumor growth paradox. And Enderling were the first to show it using a mathematical model. So now we did the same with the differential equation model, and then analyzed further with the student Jeff Bachman what to do about it. So how can we control this anomal growth of the stem cells? So stem cells are less sensitive to radiation damage, but maybe we can trick them into differentiation. And there are certain differentiation promoters like TGF-beta, Notch, and Wind, and Hedgehog, and so on, which promote differentiation of stem cells. And so if we can get them out of their stem cell compartment, let them differentiate for 2 cycles, and then we hit them with radiation, this might be a way to get them. So this is actually John Löwengroup's idea. He presented it in one of his papers to combine those, so to combine differentiation therapy and radiation therapy. And then with the student Bachman, we worked this out for head and neck cancer, for brain metastatic cancer, and for breast cancer. And I'll show you only one result here, which is this. Now this is again a tumor control probability curve. So here is treatment failure, and here is treatment success. And then on this axis, we have the radiation dose in gray. And this axis is the concentration of the differentiation promoter. So without differentiation promoter, just look along this axis, you have a typical TCP curve, which goes from 0 to 1. And so this black line, again, is where this treatment ends. And in this case, the treatment is right on. It ends where the treatment success is to be seen. So this is great. But now, for some reason, if you need to reduce radiation treatment to the patient, maybe they're more sensitive to it, then you could compensate by increasing or giving a dose of differentiation promoter, and then the curve goes up earlier. Yeah, so this is basically all I wanted to talk about. So let me summarize a little bit. I started with cancer as an ecosystem. I think this is a very important interpretation. In the glioma context, it means the microenvironment. And in particular, the geometry, I think this plays a very dominant role in glioma spread. And we tried in our model to get the geometry included. Now, for radiation treatment, several tumor control probability models are around. They are equally useful, I think. However, it needs to be, including different cell types seems to be very important. And so if looking at a tumor, one should try to understand what kind of cell types are involved. Cancer stem cells, they can explain a large number of observed phenomena, for example, the tumor growth paradox, and a combination therapy could be able to control them. Now, a final conclusion, which I have not shown actually, is the role of the immune system. So the immune system plays a very dominant role. In particular, a tumor in vivo and in vitro on a petri dish might be very different, because in a petri dish you don't have immune cells around. So I hope I was able to convince you that mathematical modeling is a useful method to understand and manage the ecosystem cancer, and we should not give up to try to do something useful with it. So here are my collaborators for radiation treatment for the cancer stem cells and for the glioma, and I thank you very much for your attention.
Thank you very much. Are there any questions or comments?
Variable functions, and they didn't quite get a good answer. Then you did that with the von Mises distribution and got a different expression. Why do you get the two different results? Yeah, sure. Can we go back to the first model where they did fit the diffusion tensor with the eigenvectors from the observation and this funny scaling function?
Okay. Let's see. So you mean this one, I guess, right?
Oh, I think the one where they did have the eigen, and this have this alpha of R, that's it, day 1823.
Yes.
And you said they didn't quite get a reasonable answer. Then you do the same idea with the von Mises distribution.
We did not, no. We don't have access to those data. So we were not able to compare.
Oh, you were not able to compare, okay.
Yeah.
Then let me ask the second question. When you actually solve the diffusion equation, say that you did a simple approximation, just like expand the divergence and then drop the diffusion altogether, then you get this hyperbolic system. Will that be any good? Or is completely hopeless?
No, it is not hopeless. So that's a good idea. I mean, you can expand it out and you get a diffusion term and an advection term. And it depends where you are on the tissue. So on the white matter tracks, it is advection dominated.
Okay.
And you would, actually we find this in numerical code as well. If you check the CFL condition, at some point you have to an advection dominated code, or you have to use an advection code, and in the other case a diffusion code.
Yeah. Thanks.
Well, two quick questions. The first one is related to the cellular automaton simulation that you showed.
Yes.
You have structured cells, right? I mean, you take into account the exterior of the tumor and the interior, which are stem cells. Have people tried to do something similar with differential equations and do some hierarchical models or structured population?
This is a very good question because, where is this with my stem cell? Here. This is a very good question because I did it myself. Heiko Enderling came to Edmonton and talked about this, and then we sat together and I said, maybe this can be done with a differential equation too. So we just got this published in the Bulletin of Math Biology.
Well, I'd ask you later on for the reference then, or is it available?
Well, it's on my website and I can show you the reference too.
And just one last question. When you build the diffusion tensor, at some point you have anisotropy, right? So you have a real, it's not always diagonal, that's what I mean.
It's not always what?
Diagonal.
Not at all, no, it's not,
So,
not at all.
So you have some sort of cross diffusion then?
Is this a cross diffusion? No, well, no, so far we do it for one species only, so cross diffusion would be between different species.
But yeah. In any case, my question is, what would be the biological, that's something that I missed, the biological interpretation of the non-diagonal terms?
Oh, no, it's just, if you have an ellipsoid, so let's say my pointer is a cigar-shaped ellipsoid, you can rotate it in any direction. And if it's not aligned with the coordinate direction, it will have non-diagonal terms.
Oh, okay.
Yeah. So, or say it the other way, you can always find a coordinate system, so it's diagonal.
When you had this advection or transport term that then you ignored, what is the physical mechanism? Somehow you mean that the tumor is, if it's on one of these little lines, it's advected or it moves. Is that what you meant, or I don't understand?
So yeah, this advection term disappears just because there's a symmetry. So if a cell is sitting, let's say, on a white matter track, it might go up or down, but the probability of going up or down is the same. So on average, it cancels.
But at the microscopic scale, there is some kind of motion.
Yes, that's right.
What is the physical mechanism for that?
That's a very good question. So the brain fibers are myelinated axons, so there's a myelinated sheet around them, and it is now known that the glioma cells do not bind to the myelin. However, between these fibers is extracellular matrix, like collagen fibers and other material, and now people think that glioma cells attach to this material to glide along. But it's very much an open question. It's not really known.
And so the scale of these fibers is the nanoscale, I imagine, or it's an atomic scale? Or
No, it is not. It is a cell scale.
Okay.
It is the same scale as cells.
So, because I was wondering when you somehow have the PD model that has to be resolved to some extent, but then you have, is there a smaller scale than, I mean, you see this jaggedness in the pictures, which reflect, I guess, the tensor, the diffusion tensor, but the diffusion tension is a scale that is much bigger than that's what I don't understand. What's the scale of variation of the diffusion tensor as opposed to the scale of these microscopic things?
Yeah, very good question too. So this is huge. The brain is, the whole brain domain has about 256 pixels. So well, if you take one two-dimensional sheet. So which means each pixel is in the range of a few millimeters, which is huge compared to the actual fibers.
In the first model, the equation had like a birth term.
Yes.
Then say that in your model with the anisotropic tensor, you add the birth term and a Fisher type term, like U
Yes,
minus
right.
U squared. Then is it relevant for these brain tumors, the fact that you can get something ahead of the main front? Does it happen, or it's absolutely nothing to do with the context?
First of all, it is certainly relevant, but we have included it in our simulations too. But it seemed to only play a role in the tumor mass in the center, but not so much in the spread in the front.
Do you know of any instance where anisotropy and this change in birth rate may play a relevant role, or it's just a mathematical thing?
No, I think it can play a very important role in different situations. So my feeling is in our situation, it's really kind of a diffusion-based spread, and it's not a Fisher wave, which we are looking for.
Okay, thanks.
Any other question or comment? Well, we thank Professor Hillen. Thank you very much.
Thank you very much.
Bien, muchas gracias por su presencia y por su atención.